Let's talk what is hematopoietic stem cell transplantation. So as the name implies, hematopoietic stem cells. So this stem cell has potential to differentiate into the mature blood cells. So what are the different types of blood cells we have? We have RBC, WBC, platelets, and WBC is of different types. So one is called granulocytes, others are called non-granulocytes. These non-granulocytes include monocyte, lymphocytes. Granulocytes include neutrophils, eosinophils, basophils, and other is natural killer cells. So these are different types of mature cells in our body. So these all cells are derived from differentiation of these hematopoietic stem cells. So how do they differentiate? So they differentiate into two lines. One is called myeloid line, another is called lymphoid line. These lymphoid line give rise to lymphoblasts, lymphocytes, T lymphocytes, B lymphocytes, natural killer cells. So initially they form immature cells that differentiates into mature cells. So these immature cells are called blast. T lymphoblast, B lymphoblast, lymphoblast. Blast means they have big nucleus, but when they differentiate, this nucleus becomes small. So T lymphoblast differentiates into T lymphocyte. B lymphoblast differentiates into B lymphocytes, and immature natural killer cell differentiates into mature natural killer cells. So these cells are derived from this lineage, lymphoid lineage, and this myeloid lineage forms RBC, platelets, and myeloblast. This myeloblast differentiates into neutrophils, eosinophils, basophils, monocytes. And what about macrophages that we have on the skin, in the liver? That is called Kupffer cell, specialized macrophage, in the spleen. So those all macrophages are derived from monocytes. So these are all derived from this stem cell. So if there is any disorder in any of these cells, either qualitative disorder, quantitative disorder that we think we cannot correct with medical management, then we can do that we can transplant these healthy hematopoietic stem cells that will form these healthy mature cells, because this one cell has potential to differentiate into all these blood cells we have in our circulation. So we can do both auto transplant and allo transplant. Auto means of self. For example, I am 33-year-old and currently I do not have any blood disorders. Blood disorder means any type, either aplastic anemia, leukemia, lymphoma. I don't have any blood disorders. But after 5 years, I don't know, I may have blood disorder, because constantly we are exposed to variety of environmental toxins, drugs, radiation. So all these produce some mutation in our DNA, and when there is some mutation that drives uncontrolled cell proliferation in any of my blood cells, then that will cause the leukemia, lymphoma. So leukemia is that uncontrolled proliferations of those blood cells. Or if I have myelofibrosis means the bone marrow is not able to produce blood cells, like aplastic anemia, where the bone marrow is replaced by fat cells, it is not able to produce blood cells. So I donated or preserved my hematopoietic stem cells in the stem cell bank. I go to a stem cell bank and I tell them to collect my hematopoietic stem cells and preserve it, because I may need it later in the future, because if I have some blood disorder I might require it. So what they do, that they give me some colony stimulating factor. Those are called granulocyte, monocyte colony stimulating factor. So what they do, these molecules, colony stimulating factor, stimulates these hematopoietic stem cells, and when the more hematopoietic stem cells are formed in the bone marrow, they spills into the circulation, and they connect me with a machine. They put one line here and they connect me with the machine and put me another line here. So all my blood goes through the machine, and what those machine will do, it will filter only the stem cells, and those mature RBC, WBC platelets will be sent back to my blood. This is called cytopheresis. So we can selectively filter out any type of cell with cytopheresis, and those stem cells are now preserved in the refrigerator in the stem cell bank. After 10 years, let's say I have acute leukemia, or let's suppose I have any lymphoma, then what is that condition of lymphoma is that there is some acquired mutation has occurred due to some insult to the DNA, and that is going uncontrolled proliferations of either B cells or T cells. Those are immature T cells, B cells, and not functioning adequately, and so B cells are not able to produce immunoglobulins. T cells are not doing its job of cell mediated killing of foreign antigens. That becomes a problem in lymphoma leukemia. So at that time, when I have lymphoma, a high risk, and it is not responding to chemotherapy, then what they do, that they myeloablate my bone marrow. They give radiation to my bones, so that all bone marrow inside the bone are killed, and they give me high dose cytotoxic drugs, chemotherapy drugs. So combination of cytotoxic drugs and radiation kills the bone marrow stem cells and all those leukemia lymphoma cells. So let's see this on this board. So let's assume this is the bone marrow, my bone marrow, and with this cytotoxic drugs, radiation, this bone marrow is now empty, and they had preserved my stem cell previously, so they will infuse me in my vein again, and those hematopoietic stem cells, they go to the bone marrow and settle it here, and they will again start differentiating those blood cells that I described earlier, and there is a chance that these new produced mature cells are now healthy. So that is auto transplant. Auto means they transplanted my own body's stem cell. So what is the advantage of this is that since this is my own stem cells, there is no chance of war between donor and recipient. When you transplant anyone's cell tissue in my body, what happens that my body recognizes this is that as a foreign and it tries to attack it. That causes graft rejection. So let's suppose you have transplanted any hematopoietic stem cells of other persons, then if there are some T lymphocytes, B lymphocytes remaining in my bone marrow that are not sufficiently killed with those cytotoxic radiation, then it will recognize that as foreign and it will attack it. So it will try to eliminate those hematopoietic stem cells, and those hematopoietic stem cells will not be able to engraft in my bone marrow, and that will cause failure of this transplantation. Or it can happen other way around. Other way around means the T lymphocytes, B lymphocytes that has been engrafted in my bone marrow of other persons, that will attack my body, because their T cells recognizes my body as foreign, and it will start attacking my organs, my liver, my GI epithelial cells, my skin. That is called graft versus host disease, because graft is attacking host. If host attacks graft, that causes graft rejection. If graft attacks host, that causes lots of problem, affects lots of organ. In chronic term, it affects almost all systems, but acutely the high effect you will see in skin, in GI epithelial cells, in liver, because there is some rapidly proliferating cells. So this was about, I told about auto transplant. Now let's talk about allo transplant. So allo transplant you can do in two ways. Two ways means, if I have identical twins, so I can transplant his stem cells, his hematopoietic stem cells, and if I transplant that, there is less chance of war between donor and recipient cells, because their HLA are similar, so there won't be fight. HLA are similar, but if I get transplant from some unrelated donor, then there is chance that I will have this war between donor and recipient cells. That's why they do HLA matching, and they try to match as much HLA as possible, and after they do transplant. So let's suppose this is, I had acute myeloid leukemia, let's suppose, and this 26-year-old female, she was healthy and she donated me hematopoietic stem cells. Let's suppose this is, now I am talking about allo transplant. So this is unrelated donor, and the doctors did HLA matching and they found that HLA is matching and they did transplant. But this is allo transplant. I don't know her. She is not my sibling or any relatives. And these hematopoietic stem cells now go here in the bone marrow, and they starts differentiating into T cells, B cells, other myeloid lines. Although my bone marrow was irradiated, given high dose cytotoxic chemotherapy, there is chance that there are still some hematopoietic stem cells of mine in my bone marrow, because you cannot eliminate it 100%. There is always somewhere hiding these hematopoietic stem cells, some myeloblast or lymphoblast. If depending upon what type of leukemia I have, let's suppose there are 50 myeloblasts in this bone marrow hiding here, and 20 hematopoietic stem cells are still here of mine, and this is of 26-year-old female, and I don't know her, and she gave me hematopoietic stem cells. So there are two different DNA chromosomes, because if you do karyotyping of her cell, hematopoietic stem cells, you will find 22 pair of chromosome with XX. If you do my karyotyping, 22 plus XY. Even these 22 pairs of chromosome, if you do karyotyping, there are different there are differences in that. But here there are no mutations or translocation that will cause me leukemia or lymphoma. That's why I accepted her hematopoietic stem cells. Now there are some antigen presenting cells here, let's say. So this antigen presenting cells, what will do, it will encounter two lines of cells. Here is her cell and here is my cells. So what it will do, it will encounter its some antigen and it will process its antigen and it will present to the T cells. So since this hematopoietic stem cells has differentiated into these T lymphocytes, so this is the same antigens are here, so it will recognize as self and it won't kill these hematopoietic stem cells. These T cells won't kill hematopoietic stem cells. If you are getting or not, I don't understand, so let me tell it again. So this is antigen presenting cells. So you know there are two lines of cells. This is my cell and this is her cell, this 26-year-old female, and this antigen presenting cells encounter some antigens on these hematopoietic stem cells and it presents to the T cells that I have found some antigens, but this T cell says that, oh, this is my own cell, so I won't kill it, so it spares and it starts going proliferating. But this antigen presenting cells again circulates here, travels here, and again it finds some antigens and this again presents to this T cells, oh, I have encountered some antigen. So it tells that, oh, I have never seen it before because this might be some foreign. So it starts attacking this. So it starts killing these myeloblasts, lymphoblasts, hematopoietic stem cells. This is the concept of graft versus leukemia effect. If the graft versus host disease is seen in other organs, that is called graft versus host disease. But if this is seen in the bone marrow, it is called graft versus leukemia effect. So what is this doing? That it is help killing these myeloblasts. If there are some hiding myeloblasts and that was not completely ablated with these cytotoxic drugs, chemotherapy drugs or radiation, then this will also help killing these myeloblasts. That's why we do allogenic transplant in leukemia, in acute myeloid leukemia, ALL. Because this graft versus host disease can be utilized in killing the remaining myeloblasts or lymphoblasts. So this is not only, transplant is not only limited to this leukemia. As I told that this can be used in all types of blood disorders that you cannot think that you cannot be cured with the medical management. Like you see pure red cell aplasia, aplastic anemia, qualitative disorders of hemoglobin like sickle cell anemia, sickle cell disease, thalassemia, major, myelodysplastic syndrome, and disorders of the WBC. You know there are a variety of enzymes in T lymphocytes, B lymphocytes, WBC, and when those enzymes are deficient, those called variety of immunodeficiency states. You know, severe combined immunodeficiency, common variable immune deficiency,